is a light-dependent reaction, and it occurs in the thylakoid. Here are the inputs. H2O, light energy, goes to ADP, and an ADP. And here are the outputs. NADPH, ATP, O2.
We're inside the thylakoid, which is inside the chloroplast for the light-dependent reaction. We begin with light energy entering into photosystem 2, which breaks apart H2O into oxygen and protons and also electrons. The electrons go down the electron transport chain to photosystem 1, which reduces NADP plus to NADPH. The electrons that went down the electron transport chain are also coupled with proton translocation, which causes protons from outside to enter in to join the protons from earlier. And this causes chemical potential energy, which causes the protons to go through the ATP synthase and cause ADP through chemiosomatic phosphorylation to become ATP.
Okay, so now we are going to learn the process of the Calvin cycle. The Calvin cycle takes place in the stroma inside of the chloroplast. So the inputs in the Calvin cycle are CO2, ATP, and NADPH. And the outputs are, the output is G3P. And the first step of the Calvin cycle is the CO2 fixation. In the CO2 fixation, CO2 combines with RuBP. RuBP is a 5-carbon molecule and CO2 is a 1-carbon molecule. So when they're combined using the Rubisco, which is an enzyme, it makes a 6-carbon molecule, which is then split into 2 3-carbon molecules. So the second step of the Calvin cycle is the NADPH reduction. So what happens is that the 2 3-carbon molecules get combined with NADPH and ATP, and they form G3P. The last step of the Calvin cycle is the regeneration of RuBP. What happens is that the Calvin cycle will repeat itself 3 times, and thus creating 3 molecules of G3P. Only 1 molecule of G3P will escape and become glucose. The rest of the 5 G3Ps will use the energy of ATP, and they will make 3 molecules of RuBP, which will be reused in the Calvin cycle.
This is the first step of cellular respiration, glycolysis. It occurs in the cytoplasm. The inputs are glucose, NAD+, and ADP, and the outputs are pyruvate, NADH, and ATP. This is the first phase of glycolysis, the energy investment phase. It's called the energy investment phase because glucose, a 6-carbon molecule, uses 2 ATPs to be broken down into 2 G3Ps, which are 3-carbon molecules. This is the second phase of glycolysis, the energy harvesting step. And in this phase, the G3P from the energy investing step is broken down into pyruvate, which is also a 3-carbon molecule, and in doing so, 2 ADPs are transformed to 2 ATPs, forming energy, and NAD+ is reduced to NADH, which carries electrons into the electron transport chain, and finally, water is a byproduct. This is the preparatory reaction, the second step of cellular respiration. It occurs inside the mitochondria, specifically the matrix. Its inputs are pyruvate from glycolysis, NAD+, and coenzyme A, and its outputs are CO2, acetyl-CoA, and NADH. The pyruvate from glycolysis is used in the preparatory reaction, and what happens is this 3-carbon molecule loses 1 carbon at CO2, and what remains is a 2-carbon molecule. And the CO2 essentially just leaves the body, and the 2-carbon molecule is coupled with coenzyme A. A to form acetyl-CoA, which is used in the next step of cellular respiration, and in this process NAD plus is reduced to NADH, which is used later on in the electron transport chain. This is the citric acid cycle, the third part of cellular respiration. It occurs in the mitochondria, and the inputs are oxaloacetate, NAD+, FAD, and ADP. And the outputs are once again oxaloacetate because it's a cycle, CO2, NADH, and FADH2O, and ATP. This is the first half of the citric acid cycle. What happens is the acetyl-CoA from the preparatory reaction comes in and bonds with oxaloacetate to form citric acid, which is what the cycle is named after. The citric acid is then proceeding to be broken down into 2 CO2s and the remaining 4-carbon molecule. And in doing so, 2 NAD pluses are reduced to NADHs, which are once again used in the electron transport chain later. This is the second half of the citric acid cycle, and the 4-carbon molecule that was broken down from citric acid eventually goes around after a few turns and becomes oxaloacetate again, the original substrate. And in the process of becoming oxaloacetate, ADP becomes ATP, and NADH and NAD plus and FAD are both reduced to NADH and FADH2, which are both used in the electron transport chain.
And lastly, we're going to talk about the electron transport chain. The inputs of the electron transport chain are NADH and FADH2, and the outputs are H2O and ATP. Okay, so the electron transport chain is located in the cristae of the mitochondria. The cristae is the folds of the inner membrane, and the electron transport chain is a series of electron enzymes that accept high energy electrons from NADH and FADH2. So when these electrons connect with the enzyme, it propels a hydrogen proton to go out to the outer membrane. And so this repeats until it goes to the very last enzyme. And at this last enzyme, the electron has lost its power. And this is a problem because we need more electrons to go through the chain, but it can't move. So what happens is that oxygen takes away the electron, and combined with the hydrogen, it will create water. So as all these protons gather in the outer membrane, they will want to go back into the mitochondrial matrix, and so they will flow through the ATP synthase, and through this, ATP will be created through the process of chemiosmotic phosphorylation. And all of these components of cellular respiration overall happen independently, and they are not linear.